Myofibrils consist of units called sarcomeres. Each red cylinder represents a sarcomere. This is a brief overview of the model that I created. The pink twists toward the top of the page represent the actin, and then the blue twists toward the bottom of the page represent the myosin. The blue circles represent troponin, the toothpicks represent tropomyosin, and then the circles under the toothpicks, or the circles that are covered by the toothpicks, represent the cross-bridge binding sites. And then lastly, the purple thing that's sort of coming out of the myosin represents the myosin heads. Muscle contractions are controlled by calcium. Regulatory proteins of troponin and tropomyosin are found on the actin filament. When the muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. However, when there is a high concentration of calcium, the calcium is going to bind to troponin, displacing the tropomyosin and then allowing the myosin head to bind to the cross-bridge binding sites. Calcium ions are stored in the sarcoplasmic reticulum and are released as a result of a nervous signal. Acetylcholine is released from the neuron. When the acetylcholine binds to the receptors, the membrane of the muscle becomes depolarized. The electrical signal then allows the calcium ions to flow to the myofibril. A contraction begins when an ATP molecule is hydrolyzed into ADP and a phosphate molecule. This causes the myosin head to extend and attach to a binding site on the actin, forming a cross bridge. An action, also known as the power stroke, is triggered, allowing myosin to pull the actin filament. During the power stroke, ADP and the phosphate molecule are released. The myosin remains bound to the actin until a new ATP molecule binds, freeing the myosin.